Flow cytometry and FACS analysis. Resting lymphocytes present a deceptively uniform appearance, all being small round cells with a dense nucleus and little cytoplasm. However, these cells comprise many functional subpopulations which are usually identified and distinguished from each other on the basis of their differential expression of cell surface proteins, which can be detected using specific antibodies. B and T lymphocytes, for example, are identified unambiguously and separated from each other by antibodies to the constant regions of the B and T cell antigen receptors. T cells are further subdivided on the basis of expression of the co-receptor proteins CD4 and CD8. An immensely powerful tool for defining and enumerating lymphocytes is the flow cytometer, which detects and counts individual cells passing in a stream through a laser beam. A flow cytometer equipped to separate the identified cells is called a fluorescence activated cell sorter, FACS. These instruments are used to study the properties of cell subsets identified using monoclonal antibodies to cell surface proteins. Individual cells within a mixed population are first tagged by treatment with specific monoclonal antibodies labeled with fluorescent dyes or by specific antibodies followed by labeled anti-immunoglobulin antibodies. The mixture of labeled cells is then forced with a much larger volume of saline through a nozzle, creating a fine stream of liquid-containing cells spaced singly at intervals. As each cell passes through a laser beam, it scatters the laser light, and any dye molecules bound to the cell will be excited and will fluoresce. Sensitive photomultiplier tubes detect both the scattered light, which gives information on the size and granularity of the cell, and the fluorescence emissions, which give information on the binding of the labeled monoclonal antibodies, and hence on the expression of cell surface proteins by each cell. And that's flow cytometry and FACS analysis. Have a great day.